now we're going to listen to Maartje Boon. She's working at the University of Stuttgart, and she's going to talk about the experimental characterization of hydrogen transport in porous reservoirs around coastlines.
Okay, thank you, and thank you very much for inviting me to present here. So I'm now at the University of Stuttgart, but before I was at TU Delft, working in the Admire Hydrogen Lab, and all the experiments that I'm going to show in this presentation were carried out in the Admire Hydrogen Lab. So I'm still involved with the experiments there. So one disadvantage of the renewable energy sources is that they are intermittent in nature, which means that some days you have a lot of wind and you produce a lot of energy, more than you need. And then there are days when there's no wind and you don't have enough energy. So one solution for this is underground hydrogen storage. So the idea behind underground hydrogen storage is that on days where you have too much energy production, you store that underground in the form of hydrogen, and then you can take that hydrogen out of your porous reservoir again on days when you don't have enough. So here you can see a figure showing the subsurface, and as you can see, next to underground hydrogen storage, there's also CO2 storage and natural gas storage. So storing gases underground is nothing new. And here I'm showing a figure of the United States, and all these circles here are natural gas underground storage fields. And you can see that there are many of them, and they've been storing natural gas underground for decades. So why would we think that storing hydrogen underground is very different? Now, one reason for that is that hydrogen has very different properties. So for example, compared to methane, hydrogen has a much higher interfacial tension, a much lower density, and a much lower viscosity. And these differences in fluid properties can lead to very different flow behavior. In addition to that, it is also expected that microbial activity will play a very important role during underground hydrogen storage, because certain microbes can use hydrogen for their metabolism, and that can lead to many unwanted effects. For example, you can get, of course, hydrogen loss, and that's not what you want, because that means energy loss. During their metabolism, they also produce H2S, which is toxic, so you'll get gas contamination, also not what you want. And when they grow a lot, they can form biofilms, and these biofilms can lead to clogging, and you get reduced injectivity, which is also not favorable. And then these biofilms can have a different wettability than your rock surface, so you can get wettability alteration. So here I'm showing an experiment that we carried out. This is a microfluidic chip, and this chip was initially filled with water or brine, and then we started to inject hydrogen. So you can see how the hydrogen with the darker black lines is displacing the water. And this very nicely mimics what happens when you inject hydrogen into porous reservoirs. But of course, these porous reservoirs or the pore structure of porous reservoirs is much more complex than this microfluidic chip. So here you see 6 different rock types, and you can see that these grains and these pore spaces are much more heterogeneous. And of course, this will have a big impact on how this hydrogen is flowing through these porous reservoirs. Now, next to heterogeneities at the pore scale, you also have heterogeneities at the core scale, the log scale, the outcrop scale, and the field scale. And of course, all these heterogeneities will have a big impact on the overall flow behavior in your reservoir, and also on the amount of trapping that you will have in your reservoir. And we want to, of course, know how these heterogeneities impact the flow behavior and the trapping during repeated injection and production cycles, and how much of the hydrogen that we inject into this reservoir will we be able to produce again. So now let's first have a look at the pore scale. So a very important parameter that determines how the hydrogen and the brine are flowing together through this pore space is the wettability. So the wettability is the tendency of a fluid to spread on a solid surface surrounded by another fluid, and it can be determined by the contact angle. And this wettability has a very big impact on the overall flow behavior in your reservoir. Because, for example, if you would have a reservoir which is gas wetting, then if you inject hydrogen, then the rock grains would like to be in touch with the hydrogen, and the hydrogen would be flowing even through the smallest pore spaces. But if your rock is water wetting, then hydrogen would be mainly flowing through the bigger pore spaces. So you can see it has a big impact on the flow behavior during injection, but it also has a very big impact what happens during production. So this wettability is a very important parameter if you want to understand what happens in your reservoir. And therefore, when people started to get interested in underground hydrogen storage, this was one of the first measurements that people were making in the laboratory. And they were using several different techniques. So you have the tilted plate technique, captive bubble cell approach, the microfluidics, which I showed on the previous slides. And you can also measure contact angles inside real rocks. And of course, those are maybe most representative. So here you see sort of a short summary of these contact angle measurements, not complete, but that were made for the hydrogen brine rock system. So here on the y-axis you see contact angle, and then the light blue bars are advancing contact angles, the darker blue are the receding contact angles, and then the dashed lines are static contact angle measurements. So from all these measurements, it was clear that this hydrogen brine rock system is water wet. But now the question is, how is this impacted by microbial activity? Or that was a question that we were interested in. So before us, there was one other study that looked at the impact of microbial activity on the wettability, and they used microfluidics. So this was a study by Liu et al. So they had a microfluidic chip, and they filled it with living brine containing sulfate-reducing microbes, and then they started to inject hydrogen. And here you can see some of their results. So on the left, you see a graph with contact angle on the y-axis and time on the x-axis. And here you see the chip, the microfluidic chip on day 0 and day 1. So what they observed was that initially the contact angle was around 40 degrees, and then during the experiment, due to this microbial activity, this contact angle increased to 100. So there was a big increase in contact angle, and the system shifted from water wet to neutral wet conditions. So now we also wanted to look at the impact of this microbial activity on wettability by measuring contact angles, and for that we use this captive bubble cell approach. So in this approach, you have a cell filled with water or brine, and then you have a rock surface here, and you release bubbles of gas here from the nozzle, and they rise due to buoyancy and then slowly dissolve into the brine. And for all these bubbles, you can characterize the contact angle. And we use this technique to measure contact angles for hydrogen and hydrogen methane mixtures. And now we wanted to use the same the same experimental setup for this living brine experiments. And therefore we had to, we made some adjustments. And one adjustment that we made is that we included a pH meter, because the activity of these microbes can be indicated, or the pH indicates the level of activity of these microbes. We also included a glove box, so the living brine was also always inside this glove box, because it cannot handle any oxygen, so it had to be handled in a nitrogen environment. And the experiment was carried out at 37 degrees and 3 bar, and we used a pure quartz slab and sulfate-reducing microbes. So the experiment lasted 39 days, And this is the surface of this pure quartz slab at the end of this experiment. And you can see that a very nice biofilm formed on this surface. So now let's have a look at how this impacted the contact angles. So here are two figures. One on the left is contact angle on the y-axis, volume on the x-axis, and here it's pH on the y-axis and time on the x-axis. And the red curve here that you see is for Bentheimer, so it's another rock type. It's not really interesting for now. And here you can see the contact angles for quartz using just DI water. And here you can see in green the contact angles for our quartz slab on day 1 when we started to inject the living brine. So here you see that it's different from DI water, but that's not due to any biofilm formation yet, but that's due to different or a lower interfacial tension. So here you see that on day 10 the contact angles decreased, and this is on day 15, and you can see that the pH is increasing, day 25 and day 30. So we saw that this microbial activity made our system more water wet, and we were a bit confused at first with these results, because we knew from these other people that did these look at the impact of microbial activity on the contact angle, that they saw a system where the contact angle was increasing and which was becoming less water wet. And we saw that it became more water wet. So now I'm not a microbiologist, but it turns out that this, the wettability of the biofilm depends on the consistency of your extracellular polymeric substance. And so that's what is created when this biofilm is formed, and that consists of hydrophobic and hydrophilic components. But the consistency of this extracellular polymeric substance really depends on the growth conditions. So it really depends on your pressure, your temperature, your pH, the strain of microbes that you're using, and of course the chemical condition of your brine. So that was after finding this out, we realized that it's very difficult to really look at the impact of microbial activity because you really need to accurately replicate the conditions of your reservoir, and that is not so easy because it's very difficult to get the right strain, or even to determine, I guess, the strains of or the colonies that you have in your reservoir, and also to really create the same conditions. So now let's look at some core scale experiments. So here you see an example of a rock core at the core scale, and you can see that this has all sorts of small scale heterogeneities, and these heterogeneities can have a big impact on your flow behavior, but also can result in a lot of trapping because they can form capillary barriers. So now we carried out an experiment using a heterogeneous rock core with capillary barriers. So this was a Berea sandstone rock core. It was 17 centimeters in length and 3.8 centimeters in diameter. We used a hydrogen water system, and the experiment was carried out at 100 bar and 20 degrees C. And here you can see the porosity map. So here in the blue you can see these two low porosity bands. And so we carried out multiphase flow experiments. So we injected hydrogen and water into this core, and then we visualized with this medical X-ray CT scanner how this hydrogen and brine were flowing together through this rock core. And we carried out a drainage experiment and also imbibition experiments. So you can see some of the drainage results. And you can immediately see that there was a lot happening, and it was very different to the results that I'm used to with working with the CO2 brine system. So for one, we saw this gravity segregation at the inlet, and then due to this capillary barrier, we saw that this hydrogen that was flowing here at the top was pushed down, and it was actually quite well spread further down the core.
Why do you have this barrier?
So that's a low porosity zone, as you can see here. So it's a very low porosity. And
like in the sandstone?
So very tiny pores.
Okay.
And
sometimes when you have, for example, 3D scans and you want to merge them after reconstruction, you have this kind of effect. I was wondering if it is the effect of reconstruction.
no, it's not. It's a real barrier, as you can also see, because the flow behavior is also impacted by this barrier. But it's just very tiny pores, which acts as a capillary barrier. So you have to overcome a higher capillary pressure to enter there, for the hydrogen to enter there. Yeah. And therefore, instead of entering right here at the top, it was pushed down and spread more evenly further down the core. So we also carried out an imbibition experiment, and so here you can see the results for imbibition. And also there we saw very different flow behavior, or very different behavior. We saw that here at the inlet, there were high water saturation fingers forming, and these high water saturation fingers evolved into a channel later on during the experiment. And then when we were injecting 100% water, so this is your trapped hydrogen, or when you've immobilized your gas phase, you can see that all the water is just flowing through this channel, and therefore bypassing a lot of the hydrogen, which led to a lot of trapping of the hydrogen. Now, if you want to make multiphase flow or measure multiphase flow parameters, you need to have your hydrogen and your water flowing through the same pore space. You cannot have this gravity segregation where you have this side-by-side flow, or this channel formation is also, you cannot really make those measurements. But we were a bit lucky because we had a setup which had several pressure measurements along the length of the core, so we could still make meaningful measurements. So here you can see the relative permeability curves that we measured. So this is relative permeability on the y-axis and water saturation on the x-axis. And in gray, you see the relative permeability curves for drainage. So this is water, this is the relative permeability for your hydrogen, and then in green and yellow you see the relative permeability for imbibition for two different cycles. And what is interesting from these experiments, and what other people have also observed, is that the hydrogen relative permeability is very low, much lower than the ones that we measure for CO2 brine systems. So we then used these measured relative permeability and capillary pressure functions, which I'm not showing here, to simulate underground hydrogen storage at the field scale. And we simulated two different scenarios. So here at the top, there is simulation where we did not include hysteresis, so we just used the drainage curves. And here at the bottom, we used we included hysteresis, so we used drainage and different imbibition curves and with different scanning curves. So let's first have a look at the case without hysteresis. So we first injected methane. So methane is here the cushion gas, and then we have 10 injection and production cycles. So this is the case where there's no trapping included. And here you can see what happens when you include hysteresis, so when you include trapping in your simulations. And so you can see that the flow behavior is very different due to this trapping, and also due to this trapping, the working volume that you have in your reservoir is much smaller. And also the amount of hydrogen that you can recover again from your reservoir is very different when you include hysteresis in your simulation. So if you don't include hysteresis, you can almost recover all your hydrogen, so 98% recovery. And if you do include this trapping, you have 84% of recovery. So now I would like to go back to this figure. So this is the trapped hydrogen saturation. So this is at the end of imbibition after injecting 100% water, and you can see that all this hydrogen here is getting trapped because all the water is flowing through this channel. So now what is interesting here is that I mentioned before that these capillary barriers and these heterogeneities, those are normally causing the trapping, but in this case it's not the heterogeneities that is causing or resulting in a lot of trapping, but it's actually the channel formation due to hydrogen dissolution into the brine that is causing all this trapping or bypassing of this hydrogen here. So you can also see that on this figure on the right. So this is residual hydrogen saturation on the y-axis and initial hydrogen saturation on the x-axis. And the color corresponds to the location in the core in this direction. So blue is here at the top and red is here at the bottom. See that so inside or outside the channel here, where the blue colors, almost all your hydrogen that was initially there is trapped. And in the channel here, the red color, almost all the hydrogen that was here is removed due to dissolution into your brine. Now this was a really big surprise to us because the hydrogen solubility is very, very low. It's, I think it's almost like 1,000 times smaller than the dissolution of CO2 into water. So when we started doing these experiments with the setup, we said, okay, we don't need to really care about hydrogen dissolution. And instead of, so with the CO2, when you do the experiments, you always have an extra flow loop to really equilibrate the hydrogen and the brine with each other. But in this case, we thought if we just have very long tubing, then the hydrogen and the brine will already equilibrate.
Sorry. Is it due to the gravity that it seems like half of the rock is more saturated than the other half?
Yeah, so part of this is due to gravity because the water was initially flowing at the bottom and because it was flowing here, the hydrogen dissolution was starting here, so these fingers started to form here. And when you have these fingers which have a much higher water saturation, so a much lower hydrogen saturation, then of course the relative permeability in those fingers for the water is increasing, so it becomes more and more easy for the water to flow through these channels, and it starts growing and eventually formed this channel. So if we would not have, if we wouldn't have had this gravity segregation, maybe it would have looked differently. And I will show you an example in a second. So back to this dissolution that really surprised us, because this is not what we expected. But we did another experiment, and we were using a very special scanner this time. So this is a scanner which can handle very large samples. So the sample was again 17 centimeters in length, but it can scan at very high resolution. So we could look at the pore scale and the core scale. So the whole 17 centimeters was scanned at this low resolution, and just this 1 centimeter in the center was scanned with this very high resolution. So we could also look at this pore scale behavior. And so we had this new try, and this time we created a bypass to make sure that before we started the experiment that we really equilibrated the hydrogen and the brine very well with each other to make sure that we would not have any dissolution. So here you can see the results. So we again did drainage and imbibition, and you can see again we had these fingers forming, but not only this time during the imbibition, but we also saw these fingers forming now during drainage. So we were really puzzled by this, or confused a bit in the beginning, because this is not what you expect. So now let's have a look at this first core. So these are all different fractional flows. So when you do an experiment like this, you inject different fractional flows of hydrogen and water, and then for each fractional flow you wait before steady state is reached, and when steady state is reached you can measure 1 point on your relative permeability curve. And this right here is our first drainage point, and we were injecting just 10% hydrogen and 90% brine. And so here we have a close-up of this core, and here you can see the pressure drop along the core during the experiment. So here is the delta P, and this is time. And you can see when we started the experiment, it was behaving like a normal experiment. So we first had an increase, and then it was a plateau right here. So we thought, okay, steady state is almost there. But instead of keeping this plateau, the pressure started to increase, and it was not just increasing a little bit, it just kept on increasing. And when we reached a delta P of 18 bars, we switched the flow rate because it just kept on going. And we were really puzzled what was happening, but now we were lucky that we also had the pore scale scans. So here you can see the disconnected hydrogen ganglia during drainage. And this is very different because normally during drainage everything is connected. But what we saw is that we have all these disconnected ganglia during drainage, and we think what is happening is that we have all this hydrogen dissolution happening at the inlet, and further down this dissolved hydrogen is coming out of solution and is forming all these disconnected ganglia, which then really blocks the flow and leads to this very high pressure buildup. So it had a very big impact on the whole system. And of course this was just at the inlet, and also here. Because you answered, or you asked, okay, is it due to this gravity segregation? In this time, we had the core vertically, and you can see that even there we had these fingers forming. So now, of course, this is just at the inlet, but you can imagine if you have a more heterogeneous core, you will also have pressure variations within your core, and it seems like hydrogen very easily goes in and out of solution in the brine. So these could be that due to this capillary heterogeneity or heterogeneity in your core, you can have these processes happening also at different locations. So this dissolution is a lot more important than we thought. And another process where dissolution is important is Oswald ripening. So after this first fractional flow, we stopped the flow, and we stopped it for 39 hours. And we scanned it at the start, initially, and then we scanned it again at this high resolution after 39 hours. And so due to this process of Oswald ripening, you get a redistribution of your hydrogen. So what happens with Oswald ripening is that when you have smaller hydrogen ganglia, they have a higher capillary pressure, and because they have a higher capillary pressure, more hydrogen can dissolve. And then when you have bigger hydrogen ganglia, they have a lower capillary pressure, so less hydrogen will dissolve around there. So therefore you can get these concentration gradients in your hydrogen saturation in your brine, and you get this flow from the small bubbles towards the bigger bubbles. And you get that these small bubbles are disappearing and your bigger bubbles are growing. And you can also see that here. So this is the image initially, and this is after 39 hours of storage. And here the ganglia sizes were, divided into small, medium, and large sizes. And the small are in blue, the medium sizes are in green, and the large sizes are in brown. So initially, here you can see, if you look at this purple circle, you can see that there is this mid-size ganglia. And this mid-size ganglia after 39 hours is, came apart into smaller bubbles. So some of it has dissolved, and you got these smaller bubbles here. And you can see that this green, this mid-size bubble here, now turned into a large size bubble, so a brown color here. So also here, this dissolution. So hydrogen dissolution is much more important than we thought. So to summarize, underground hydrogen storage could possibly provide large-scale energy storage for the energy transition, but there are still many things unclear, and this hydrogen brine rock system is not yet well understood. And in this presentation, I looked at the impact of microbial activity, which is very difficult to really investigate, I think, and also the impact of hydrogen dissolution into brine seems to be important to further investigate. So thank you very much for listening, and I'm happy to answer any questions.
Great, thank you so much for the presentation. And now, do you have some questions? Yes?
Good interesting. Yeah, thanks very much for that. I had a question about the field scale simulations.
Yeah.
And so I know there's this concern about the cushion gas, and so I guess here there's a kind of a natural gas being used as a cushion.
Yeah.
And I was wondering how much of an issue or an uncertainty is the mixing between the hydrogen and the natural gas playing a role here, and is it difficult to estimate?
So in this case, we didn't really pay attention to that, so I cannot really tell, but it's one of the big uncertainties as well for underground hydrogen storage, and there are many people looking into that and quantifying that, and therefore people also say, so you can, some people say, okay, you should use CO2 because it has a very high compressibility and therefore it's a really good cushion gas. Other people say, why don't you just use hydrogen, because then you don't have this mixing and you don't have that problem. So there are many people looking into different cushion gases and yeah, also into this mixing problem.
I had a very simple question. I was wondering what are the pressure and temperature conditions in your core? Are they exactly relevant to the
No, because we had 100 bar and 20 degrees C during this first experiment. The reason why we did that was that adding the temperature to the experimental setup was another complication to add to the experimental setup, and to get these experiments running, or to get permission to run these experiments with hydrogen, was already not so easy. So we didn't worry about the temperature. But yes, that's important too.
So with a higher temperature, the solubility will be even higher.
I think so. Yeah. So but we didn't look at that. And this last experiment was carried out at 50 bar. And I think, what was it, 23 degrees C or something like that. So also not really pressure, reservoir conditions, but high pressure, but not high enough maybe, and temperature also not high enough.
Thank you for the great presentation. I was curious and a bit surprised by what you said about the exclusion of hydrogen to the point of forming ganglia that would block the flow of water. So the solubility of hydrogen is
very low.
So how can you have enough hydrogen to exhaust to the point of filling in entire pores?
Well, so when we do these experiments, let's see if I can go there. So when we do these experiments, we run it for many pore volumes. So we do a lot, and I think little bits get stuck. And initially, it was just behaving quite normally, and I think it just gets more and more. But this is also it's a hypothesis. We haven't really tested it. Also, this is so the resolution here. I think it's 6 micrometers. It could also be possible that the connections are just very tiny, that you don't see the connections, but there are actually connections. So that is also something we don't know yet, but it's just that there is no way we can explain, or we don't know why we see this behavior and these very extreme pressure drops, because a pressure drop of 18 bar for an experiment like this is really out of what you expect.
Yeah, just for the pressure drop, if you're having such a high pressure drop for your, I think this benthammer sample, if you have really heterogeneous samples, then it would be then impossible to do any kind of reliable measurements in the lab. Because we generally expect to have high pressure drops even in CO2 for the heterogeneous rocks. So is there any kind of strategy how to proceed with more heterogeneous rocks like carbonates?
Well, I haven't thought about that. I think the problem is, so when you equilibrate your brine, you equilibrate it, of course, at conditions that you want to carry out your experiment. So in this case, it was 50 bar. So your brine is equilibrated at 50 bar. But of course, when you start to inject, you always get a pressure buildup. So you could maybe say, okay, instead of equilibrating at 50 bar, I expect to see a pressure drop of this much. So I equilibrate at higher pressures, and I start to inject that, something like that. I don't know, but we haven't really.
Well, with this, thank you again for coming.